The following is a short film on aseptic refeeding technique for an adherent cell culture. Both the good and poor laboratory practice techniques will be illustrated. The first scene will illustrate proper personal protective equipment. Notice Laurel on the left donning her white lab coat. Mike on the right is removing his shoes, which is improper. Notice that Laurel has the proper closed-toed shoes. Mike is not supposed to be drinking in the laboratory. Laurel's hair is fastened in back. Laurel is now putting on the correct hand protection, blue nitrile gloves, whereas Mike is putting on his street gloves. The next PPE is the eye protection. Laurel's are appropriate, and Mike's is missing. Laurel looks excellent and safe. Mike is texting and is in danger of harming himself in the laboratory. Laurel enters the lab and will begin by preparing her work area. She has turned on the laminar flow hood and raised the sash to the proper height. She will now begin to disinfect her work area, one of the steps to avoid contamination of her cell culture, wiping it down with a 70% ethanol solution. She will then remove her media from the 37-degree water bath. Noting that the water bath water is a source of contamination, she will rinse the media container with a 70% ethanol solution. The necessary equipment is placed in the laminar flow hood. Note that only the necessary equipment is placed and not extraneous items. The final step is to retrieve these cells from the incubator, tighten the cap on the flask, handle the cells gently. Laurel performs a visual inspection first to look at color of media and any possible contaminants. Then she performs a microscopic inspection to gauge cell health, confluency, and any possible contamination. As always, the final step is to bring the cells into the hood. Laurel applies 70% ethanol to her gloved hands. Again, a step to avoid contamination. And just to be extra safe, she wipes down the surface of her hood one more time with alcohol. Mike will illustrate what not to do. First, he enters class late, talking on his cell phone, not with the correct PPE. He raises the sash too high on the laminar flow hood and begins to insert many unnecessary items into his hood, many of which are not sterile. He then goes over to grab his cells, still talking on his cell phone, shakes the cells, which you should not do, to perform the visual inspection, and does not perform the microscopic inspection. He then turns on the light and the laminar flow hood. Mike performs one of the worst steps by opening the container outside of a sterile environment. The container flask should now be considered not sterile. In refeeding, the old media needs to be removed. Yes, Mike is drinking in the lab. And instead of aspirating the media, Mike pours the old media into an inappropriate container. Mike then needs to refresh the media. Notice the centrifuge tube is upside down. Mike removes the stereological pipette, but touches the tip, blows into it, thus contaminating it again. Touches the tip to the waste container, puts the cap down improperly, rests the serological pipette horizontally, which should never be done. Inserts the media. Notice below his left hand he has spilled a quantity of media. Mike then disposes the serological pipette in the improper waste container. And Mike is, of course, texting in the hood, which should not be done. He then cleans his spill with an inappropriate towel using no alcohol and disposes it in the improper container. Notice that Mike has the glass pipettes on the hood vent. He returns his cells to the incubator without loosening the cap and setting them upright as opposed to horizontal. Instead of cleaning the hood and properly disinfecting it, Mike simply leaves. Laurel will illustrate the proper aseptic technique for refeeding cells after turning on the vacuum pump. She must disinfect the vacuum hose, dipping it into a 70% solution of ethanol. Then wiping down the length of the hose that will be contained in the laminar flow hood with the 70% ethanol solution. Disposing her paper towel in the proper waste container. She gently removes a sterile glass pipette, connecting it to the vacuum hose. Notice how she holds her cap with her 2 fingers and aspirates the old media. Immediately upon aspiration, she recaps her culture flask. And disposes of the glass pipette in the proper trash receptacle. She then gently loosens her media container, unwraps the top portion of her serological pipette. Laurel is careful not to touch the tip of the pipette to any surface, except for the media. She draws up the required amount of media. Keeping the serological pipette vertical, recaps the media and dispenses the media into the culture flask. Culture flask is capped and the serological pipette immediately disposed of in the proper container. Laurel then is sure to note the date, the action performed, in this case refeeding the cells, and the type of media the cells were refed with. She then replaces the cells into the incubator, being sure to loosen the cap on the flask and setting the culture flask horizontally. Laurel then proceeds to properly leave the laminar flow hood by removing the items that she used, noting that she did remove the sterile glass pipettes and she will discard of them later because they are no longer sterile. The media is returned to the 37-degree water bath for use by another student. The surface of the laminar flow hood is once again disinfected with a 70% ethanol solution. The ethanol is removed to be placed in a flammable cabinet if no longer in use. Laurel lowers the sash and turns off the hood.
That's that.